Hi, my name is Rishi Gelu, and my project focuses on understanding how structural changes in cell surface receptors can contribute to drug resistance in cancer immunotherapy. Many modern cancer treatments rely on monoclonal antibodies that are designed to bind very specific receptors on the surface of cancer cells. These therapies can be highly effective, but over time resistance often develops. One reason for this is that cancer cells can modify the structure of the cell surface receptors, making it more difficult for therapeutic antibodies to bind. One common way proteins are modified is through glycosylation, a post translational process where sugar molecules are added to specific amino acids. Glycosylation can change the shape of a protein's surface or physically block binding sites, potentially preventing drugs from working as intended. The challenge is that many cell surface receptors involved in cancer do not have experimentally solved full length structures, making it more difficult to study these structural changes directly. In this project, I explored whether AI based structural modeling could be used to predict how glycosylation alters receptor structure in a way that reduces drug binding. While this question applies broadly to plasma cell surface receptors, I used GPRC5D, a clinically relevant immunotherapy target in multiple myeloma, as a representative case study to test this approach. Because no experimentally solved full-length structure of GPRC5D exists, I first predicted its three-dimensional structure using AlphaFold through CollabFold, an AI-based protein structure prediction platform. I evaluated the reliability of the predicted structure using PODDT confidence scores, which estimate how confident the model is in each amino acid's position. I then identified potential N-linked glycosylation sites and performed solvent-accessible surface area analysis to determine whether each site was exposed, partially exposed, or buried within the protein structure. One buried site was intentionally kept as a negative control. Next, I generated glycosylated receptor models using CHARM-GUI and analyzed antibody binding using haddock docking simulations, comparing predicted binding across unglycosylated and glycosylated models. A major strength of this design is that it combines AI-based prediction with controlled structural comparisons. A limitation is that docking simulations estimate relative binding trends rather than actual experimentally measured binding affinities. The results showed that glycosylation at surface accessible residues reduced predicted antibody binding affinity, while glycosylation at a buried site produced minimal effects. One unexpected outcome was how clearly the buried site behaved as a negative control, demonstrating that structural context is more important than sequence alone when predicting glycosylation effects. The real world significance of this project lies in its potential application to cancer immunotherapy and drug development. Antibody based treatments are increasingly used to treat cancers such as multiple myeloma, but resistance remains a major clinical challenge. This project demonstrates how AI based protein structure prediction can be used to study resistance mechanisms at the molecular level, even when experimentally solved structures are unavailable. By predicting how glycosylation alters receptor structure and reduces antibody binding, this approach may help identify resistant receptor conformations before they arise clinically. In the future, this framework could be applied to other plasma cell surface receptors or therapeutic targets to guide the design of next generation antibodies that account for glycosylation related structural changes. Additionally, predicted glycosylated receptor structures could be used to screen or optimize small molecules or biologics that remain effective despite these modifications. Overall, this project highlights how AI driven structural modeling can complement experimental research and support more efficient targeted drug development strategies. I would like to thank Dr. Hamid Rezashay, Mrs. Hallie Flayhaven, and Mrs. Rebecca Keeler for their guidance and support throughout this project. Thank you for your time.